All right, everyone. So today it's a double dose of cell division. I'm going to be discussing mitosis and meiosis together because they're super tied together, and we're going to go into more depth with mitosis, but we'll do some meiosis stuff. But honestly, meiosis is a lot like mitosis, just twice. So we need to start at the beginning, though. And the previous lecture was on the cell cycle. So we're going to start there. This should look familiar. I took this from the cell cycle slide set. These next two slides are actually from that slide set. And what's over here, I just took the whole slide, so don't worry about this stuff. But we talked about cyclins and cyclin-dependent kinases being responsible for driving the cell cycle. And we're going to see that proceed and drive us through the second growth phase, which is actually probably a lot smaller than this, into the M phase, the mitosis phase, okay? So we do have a checkpoint that occurs here, and this junction here is called antiphase, A-N-T-E-P-H-A-S-E. I have that in another slide in just a second. But notice that we do have cyclin B and CDK1 proceeding into a large chunk of the M phase. That is not unintentional. Okay, it's not like they just had the same length and they just stuck it on there. So we're going to see that come into play here for one of the checkpoints. Okay, and how we actually get separation of our chromosomes. And so we just need to keep in mind what's happening. And why am I talking about this? Because I want to talk about entering this M phase and this anaphase checkpoint that actually occurs here. So these are the checkpoints that I talked about. We've gone through G1S, where we double our DNA. We've gone through our second growth phase. We've double checked our DNA all these times. We need to do one last double check, or like quadruple check on our DNA structure. And then we're going to head into mitosis. And we do have a checkpoint there to make sure that everything is lined up during metaphase. And so that's what's going to happen. So let's go into it. That anaphase checkpoint, it looks super duper similar. So if I just back up real fast to this checkpoint here is the one that's focused on in this diagram. Looking at P53, P21, you see a lot of Ps here. These are all tumor suppressors that can stop the cell cycle, and they're influenced by DNA damage or cell damage. And basically you're looking at trying to either fix the problem. First, you look for the problem. If you see one, you try and fix it. Or if it's really, really bad, then you just need to kill the cell, because why would you make a cell that's all broken? And so that same kind of checkpoint occurs at antiphase. So antiphase is this junction right here between G2 and M. And it's really, really similar. We're going to see a lot of the same proteins. There's really like only two, arguably three types of checkpoints that occur. The only real two checkpoints are this type of checkpoint, which looks for damage and then is influenced by tumor suppressors. And then, well, I mean, I guess there's three. So then we also have our checkpoint during the S phase, which I talked about more in the cell cycle. I had a whole slide devoted to that right at the end. And then we also have our M phase checkpoints, which I'm going to talk about in a little bit, where we're talking about our kinetochores actually connecting so that we can do that separation. So those are the three types. But this is honestly the biggest one when we see checking for DNA damage. And that's basically these guys' responsibility. Of course, these pathways are far more complicated, but when we see ATM and ATR, they're looking for damage to the checkpoint. So you're looking for single nicks or double-strand breaks, or things just caused by random stuff, thymine dimers, things that you can fix using a variety of techniques that we will talk later in this class. But you can see here, these pathways proceed down to tumor suppressors that then influence other tumor suppressors, and then are going to affect, here's our cyclin B and CDK1. So we come back to this. So cyclin B and CDK1, they're still here. And those are going to control whether or not we can proceed on into from G2 to M phase. And so if we block them, basically, then we can stop this from proceeding. But if we allow them to do their thing, then they will allow us to proceed, or however I say that correctly. You get what I'm saying. We have to get through this checkpoint to check for DNA damage, and then we can go on. Okay. Now, mitosis is one of those interesting things because we cover it in a lot of courses. Very early on, you're exposed in like Bio 200, and in botany, I used to teach, I think, some mitosis, at least under the assumption that you had seen it before. A lot of high school biologists talk about this because it's pretty straightforward. It is important to understand mitosis. Now, this is an upper division biology course, so I'm going to assume that you've been exposed to mitosis before. Now, we're going to cover at least for mitosis, the main steps. Okay, but I'm going to try and go into some more depth to just kind of expose you to the complexities of how mitosis actually works on more of a molecular level, since I'm more of a molecular biologist than a not molecular biologist. So, like a macrobiologist. And so we need to talk about mitosis, and I will kind of cover all the big stuff that I think you need if you're going to take the MCAT. But we're also going to go into more depth too. Just if something tickles your fancy, then you can dig a little bit deeper. Now, this is just a different picture that I have of the cell cycle. I like this one a lot because it shows all the different phases. It's just kind of backwards from the other one, which is confusing. But so you're going to go this way. We go through G1, we go through the S phase, G2. It's telling you everything that's happening, and it's got a lot of detail here. It's from somebody on made it on Wikipedia and put it up there. And then we're going to go through all the phases of mitosis. And it has all of them on here, which is really cool. It even has like pro-metaphase. But I like this one a lot. And we're going to go through each of these steps, okay? But the main goal of mitosis is to take one parent cell and split it. It is the method by which we can perform asexual reproduction, which most cells are undergoing asexual reproduction, right? There's no sex involved. We're not having an interchange of genetic material to create new cells that have a different combination of genetic material from parents, or from your parents. But in meiosis, of course, we do see that, and so we need to talk about that. Before we get into too much detail, I apologize for the quality of this, but this is honestly the best diagram I could find that covered like all the big words that I wanted on there. And so we need to talk about chromosome structure, because in this case, we're going to be seeing this a lot. We just need to understand chromosome structure, and we're going to talk about kinetochores and the centromere as well a lot. The only things that I added on here are the P and the Q arms. So when you have a chromosome, if you could take a chromosome in, say, like the G0 phase or the GAP1 phase, it would look like this if you condensed it down. Now, they're not normally condensed. We don't condense our chromatin until we actually enter prophase. But it's hard to depict that kind of thing, which I actually like. I'm going to be using the Khan Academy pictures. They actually sort of show it uncondensed, which is great. But if you could condense it down, there it is. So, and this is just one single chromosome. So you would have one from dad and one from mom. So there would be two of these. Those would be homologous chromosomes, okay? When you go through the S phase, you actually double each chromosome. So I would have one of these from mom and one of these from dad, or however I'm saying it. So you would have a second one from dad, okay? And each of those would have sort of two chunks to it. You have sister chromatids. So when you have two of them, you have sister chromatids. This only occurs after S phase. Okay, great exam question that I've seen many times. So what do we see here? Just the basic structure of it is pretty straightforward. You have a short arm and a long arm. The names are P and Q, just to make it confusing. So the P arm is the short one. The Q arm is the long one. We've got after S phase, we've got one chromosome. It's still connected at your centromere. So the centromere is more the components and the genes contained within the region of these chromosomes that are connected between the center between these sister chromatids. I'm sorry. The kinetochore are the proteins associated with that region, okay? So if you want to keep it separated in your head, the centromere is more DNA and histones and those things, and then the kinetochore is the actual structural proteins associated with basically where we're going to attach our spindle fibers to pull these things apart. Okay, so that's the kinetochore. Sometimes you'll see those things used interchangeably. They're not used interchangeably. The kinetochore is those proteins and the complex of proteins there. The centromere is more, honestly, this region is just called the centromere. And you have to remember, we have a lot of carryover from when we were able to look at chromosomes long before we could like really zoom in and see what even a gene was. So we didn't know what that was, but we did know the structure of chromosomes, because when they condense down, you can see them under a microscope, which is really cool. And you can do karyotyping and things like that. And we're going to talk about that when we talk about chromosome abnormalities, which is one of the last things we're going to talk about. Now, we're going to now move on. So there's our chromosome. Okay, we're going to see that a lot. And we're going to go through each phase, okay? We'll be using the Khan Academy pictures. They have such good stuff. And their pictures are awesome because it's not that I can't find better pictures, it's just that they're nice and consistent, and they're separate pictures, like individual pictures, and they're really high pixel counts, so they look really nice on my PowerPoint. As opposed to some of them, you get like a lot of mixed bags of like, this picture's got really good detail, but it's hard to see. So I'm going to use theirs because they do a really good job. And if you want to jump to just Google Khan Academy Mitosis, and they have an awesome set of things. Basically, a lot of what I'm going to be telling you, but I'm going to try and go into more detail. So up here, we're going to keep track of where we are, and we're going to slowly move this way, of course. And then I'm going to go through most of the stuff of what I think is important for each step, okay? Now, prophase is an interesting one, right? So we're going to do prophase, metaphase, anaphase, telophase, right? PMAT. If you need an acronym for it, or there's something about like peeing on a mat. I don't know. I've just memorize it. I don't know. Prophase, metaphase, anaphase, telophase. It's one of those things that just pops out like kingdom, phylum, class, order, family, genus, species. You just kind of have to have it stuck in that memory bank of yours. Now we're going to condense our chromatin. Okay? So they're kind of showing that here. I love this because they're showing it kind of loose and it's starting to condense, and then it's going to be more dense in the next picture. And so I just like that a lot because that's a really important subtlety. We're also going to start breaking down the nucleolar subcompartments. So when you take cell biology, which is a class I also teach, we learn about all these subcompartments in the nucleolus. So the nucleolus is a nuclear subcompartment that is responsible for basically for ribosome RNA, which ribosomes are made of proteins and RNA for structure. And so that RNA is created in the nucleolus. Now, there are tons of nuclear subcompartments. The nucleolus is technically just one of those. There's at least, off the top of my head, probably like 6 or 7 that I could think of that are not just the nucleolus, but other things. And but we're not talking about that today. So those dissolve. We don't have those separations because we basically need all the space that we can to actually condense our chromosomes and move them around and do all that fun stuff. So we'll have to rebuild all those things. And something to keep in the back of your mind is there are proteins, and we're going to get into some of the pathways here. There's constant protein dissolution and reformation going on constantly. There's so much activity going on. It's an amazing feat that we can even do this. And we just do it without even thinking. It's great. Now, so you're going to break down nucleolus. These are just the things that typically get thrown around. That's why I'm focusing on these. You get spindle fiber, polymerization, and centrosome activity. I'm going to spend some time actually talking about that step because it's really cool and it's kind of interesting. And I learned a lot actually studying this because I was not as familiar with actual spindle fiber formation. But so the spindle fibers are where you're going to actually, we're going to attach our chromosomes to the spindle fibers, which have a ton of different proteins and structures, and the centrosomes, and we're going to use those to pull our things apart. Okay, that's eventually what we're going to do. So we're condensing our chromatin down. I want to make sure that I acknowledge the term homologous chromosomes. I already talked about it, but I want to emphasize it again. So in this case, we have 2 pairs of homologous chromosomes, and thus we have 8 sister chromatids. So we've got a chromosome here. Let's say this is from dad. And we've got a chromosome here that's from mom. Okay, this could be, let's say we're in a human, this is chromosome 1, and here's a chromosome from dad, let's say this is chromosome 6, and there's chromosome 6 from mom. And we've gone through the S phase, so we have 2 sister chromatids that are identical, okay? So, but chromosome 1 here and chromosome 1 here, they have these same gene locations, right? So if you had a gene for eye color, it'd be in the same spot. If you have a gene for hair color, it'd be in the same spot, and for whatever gene. But those genes aren't necessarily identical, just their locations are identical, right? So the idea of having one from dad and one from mom that come together, okay? And then we have a copy from dad, copy from mom, and then we have multiple chromosomes, right? Humans have 23 pairs, right? All right, 23 of these, if this was like 1 and 6 and 9 and 12 and whatever, from dad, and 23 from mom, so 23 pairs, 46 total. Now, so that's prophase, things condense down, we're getting things started. A lot of times, it kind of depends, so some people will skip this next step, or they lump it in together with either prophase or metaphase. It's kind of in between the two, so we call it either late prophase or pro-metaphase, or I guess you could say early metaphase. It doesn't really matter. this is what it's usually. A pro-metaphase is what you'll usually see it listed as. And that's where you're starting to do more stuff. So you have to start breaking down the nuclear envelope because you need to have your chromosomes readily accessible and able to be pulled apart. You need lots of space. Okay? You don't want them sealed up inside that double membrane. You're going to finish condensing now, and then you're going to actually start binding your spindle fibers, or the ends of your spindle fibers, to the kinetochore. So remember, we've got our kinetochore here. That's the protein complex, and we're going to attach basically the cell's cytoskeleton and a bunch of components of the cytoskeleton onto these to actually help pull them apart. So let's talk about that. This means we need to talk about the centrosome. So you can see it on here. These are our centrioles, which we call together a centrosome. And what that is it's a scaffold. Here's my definition of it. It's sometimes you'll see it listed as an anchor. I like to think of it as a scaffold for your microtubule. And nucleation is a word that we use for when we have we're forming longer microtubules. So they're nucleating, i.e. they're like coming together. Polymerization is another term that's probably a better term. But nucleation just means like coming together. And so what you see is, so this is our centrosome, and you can see all of these this is just a depiction of all the microtubules from the cell, the cytoskeleton coming together and binding to, you basically need somewhere central to stay organized and act as a point to move out from. So your cell has a mother and a daughter centriole. Okay, those are the key two components of the centrosome. Okay, so you have these two things here, and they're just going to act as a nucleation point where you're going to initiate the growth of the spindle fibers, as we usually call them. And so so you have a mother and a daughter. This is so confusing. So there's a mother and a daughter, okay, centriole. So there's the mother and the daughter. They're usually listed like that. I don't know why. And then what we're going to do is during the S phase, you actually double them. Okay, so you end up with two of these. So I was going to go like this. So now I have two. Okay, and then they're going to go to either side of the cell, right? So we're kind of inside the cell, and this one's going to go over here. This one's going to go over here. And then when we actually finally, split the cell in half and go down the middle here, then this one's going to stay in one of the daughter cells, and this one would stay in the other daughter cell. And we get this back. Okay. And so hopefully that makes sense. And then what ends up happening is now I have a mother centriole and a daughter centriole that are now in this new cell. And then those split apart and each become a new mother, and then you make a new daughter centriole. Don't ask me why they do it that way. It's really confusing. If you're super into studying this kind of stuff, it makes sense, but I don't know. I think it's a terrible idea to say mother and daughter, but whatever. Initial versus secondary, I don't know, whatever. But that happens during the S phase. Now we need to talk a little bit about this. So here's our actual centriole here, and you can see the structure. So why did I put this on here? Well, I wanted to be able to zoom in a little bit. So here's the physical structure. This isn't just a bunch of randomness here. So we have chunks of three. Okay, so you can see three fibers. Okay, so there's 3 and 3 and 3 and 3 and 3, and those kind of wrap around each other to form this little tube for your actual centrioles. Okay, so if you look here, so here's our 3, 3, 3, 3. If you dig real deep and try and study these things, turns out it gets real complicated real fast. Oftentimes we will list those 3 as our microtubules, which you'll hear that referenced along with like actin and microtubules and other things that I can't think of in the cytoskeleton. There are so many proteins associated with the cytoskeleton. It's not funny at all. It's amazing, actually. These are just the ones listed. Like you'll see a bunch of question marks here. We don't know if that's part of it. Kendron, what is that? Composition unknown of the internal cartwheel. Okay, so there's a lot of unknowns. I've just put this on here to show you that things are complicated when it comes to spindle fiber formation. But what we really care about are either these yellow things or, in this case, these giant blue things forming these long spindle fibers. Okay, they're spindly and they're fibrous. They're really long. We have to grow them. And how you grow them, there's a whole other set of things for that. And what's also really interesting is how we can connect them to the external structures. So flagella, which is sperm are flagellated, or we can connect them to injectosomes, which is something in certain bacteria have if they want to inject or suck up things from eukaryotic cells is usually where you see it. So like E. coli, if they're really pathogenic, can use like this crazy looking injector. It looks like a little stabby thing, and it stabs it in there and can inject things that will damage the cell, and then it can like suck up nutrients and stuff. It's super creepy and kind of awesome. But what does this have to do with anything? Well, you can see the fiber construction basically of flagella and injectosomes can connect to the actin and the cytoskeleton and the microtubules of the cell. So you can see here's your centriole, and that's kind of just how it's associated. So I just thought that was interesting. Whatever, we're moving on. So we're going to assume that we're making our, we've got our centrioles, and they're going to make those spindle fibers. Our microtubules are going to come out, and we're going to follow this all the way to the end of it, where it's going to actually bind onto our chromosome, right? So here's our chromosome, and this should look familiar. This is a slide from the previous lecture that I gave on the cell cycle, where I go into an odd amount of detail talking about the M-phase spindle checkpoint. So this is where that checkpoint is going to come into play, because we need to make sure that this is the end of our microtubules coming out of here, right? So they're going to keep growing until they actually bind. We have to make sure that they're bound to the kinetochore, right? So here we can see this is the kinetochore complex, if you want, and then here's your centromere. Okay, so there's centromeric chromatin, and if it's not bound, then the centromere and the kinetochore complex is given access, or you give other proteins access to it, and they can disable this process from even happening. Because if I don't have my microtubule attached, well, then why in the world would I want to pull this apart? Otherwise, you're going to get non-disjunction. Okay, so that's how you get like trisomy 21 for like Down syndrome, or you can get trisomy 18 and trisomy 13, I think. I think those are ones where you can have offspring that survive. You can actually have trisomy of any of your chromosomes, but most of them result in termination of pregnancy earlier on during pregnancy, but those are the three that they can survive. Though I think 13 and 18 are pretty rare. They don't live long, but we've all met someone with Down syndrome, and so that's trisomy 21. And if you have a bad M-phase spindle checkpoint, that's where this could go wrong. And you can see if you have a problem with any of these things, then that could break that. And thus, with age, you actually get a breakdown of some of these things, and that's where age is correlated with your offspring having susceptibility to suffering from Down syndrome. Anyway, that is our checkpoint there, so that's super important. So that's what's happening at this point. So we're going to assume that our microtubules are connected to our kinetochore. We're going to keep moving on. We're going to head now, we went from prophase, now we're actually in metaphase. And metaphase is probably my second favorite of the mitotic phases, because it looks cool. Anaphase is my favorite because they pull apart. And we're going to talk about this stuff ad nauseum, so get ready, kiddos. During metaphase, the key is that we have the lining up of sister chromatids. Okay, they line up along this nice, we call it the metaphase plate. Okay, and you got our centrioles, or your centrosome, which is comprised of centrioles, among other things, on our ends, and they're on opposite ends. Okay, and so this is our polar, so we're polarizing the cell, right? So these are our poles now. They almost literally look like poles, and we're going to line them up, and our chromosomes, we need to get them lined up first, and then we're going to pull them apart, okay? So you can imagine there's a lot going on here, right? It's really complicated. So the lineup, to actually physically line up like this, is really, really important. We need our sister chromatids on each side. So you need your kinetochores all lined up, and basically you're getting an even physical push-pull from both sides of your kinetochore microtubules, and that's what, if you push evenly from both sides, that's what physically causes them to line up. So that's what's actually happening. And then once they're lined up, we're going to have anaphase occur. Now, anaphase is awesome, so we're going to pull apart those sister chromatids, right? And we're going to spend some time on this because I fell down the rabbit hole of anaphase and how it works. It's going to be awesome. So this is a really complex thing. Well, so I was trying to anticipate. somebody's going to ask a question about this, right? They're going to say, well, when does it know to start anaphase, and how is it actually happening? Well, that's a great question, Timmy. So it's initiated by a complex. I said a complex set of proteins, but what I really mean is a complex of proteins, which is complex itself. It's a complex that is complex. And so the actual pushing and pulling is controlled by a few different things. So there's two things I want to talk about. There's Anaphase A, which is where we actually initiate anaphase, and then there's Anaphase B, where we actually have the separation occurring. And so when you dig a little deeper into some of the subtleties of anaphase, you're going to encounter that kind of nomenclature where you have basically it's split in between A and B. A being the first one and then B being the second half. The actual pushing and pulling is controlled by a variety of kinesins and dinins, which the kinesins are going to look super familiar because you've probably seen a video with them in it. So well, let's get to it. Okay, so how do you actually initiate it? So during anaphase A, you have the anaphase. Hang on. I just paused this. You wouldn't even know that. Why did I say hang on? It's not like you have to hang on. The anaphase promotion complex or promoter complex, because it's promoting anaphase. So thank you, Khan Academy, again, for making really simple diagrams. I had way more complex ones that I was going to show you, but this one's nice and straightforward, so I like this. Something that if we have not talked about it, if you're in biochemistry at the same time as this class, we have not talked about, we're about to talk about proteins. We have not talked about ubiquitination, but that's basically how cells mark proteins for destruction. Okay, so that's what ubiquitin is for, and in this case, the APC is a ubiquitinase, and it ubiquitinates securin. So securin is basically preventing anaphase from starting, and so you need to promote the creation of this thing to get ubiquitin added to this thing, which then gets degraded, right? So there it gets chopped up, broken down, look at that. And that releases saccharase, which can then go in and then break up cohesin, which is one of the proteins holding this together. OMG, there's a lot going on, I know, but it's actually pretty kind of cool, right? So you have to get rid of this thing to separate this thing, which then can get rid of this thing, which then lets you separate them. Okay, so that's just the initiation now. Once it gets initiated, well, that process is really interesting. And so if you've ever seen the video, let me pull it up here. You may have seen this before. It's really common in some really good basic cell biology videos that get thrown around. So this is one version of kinesin. It's a motor protein, and it's for transport. So here's our microtubule. And you can imagine a lot of times we just say, oh, there's like a vesicle or whatever that's getting moved. But this could be a chromosome. Okay, same idea. And so that's what we actually see occurring here. So here are, there's a bunch of different kinds of kinesins, but you can see some of them are going to be pulling the chromosomes. So we assume that the chromosomes are sort of attached here. So there's two things happening. We attach the sister chromatids, one side to a kinesin, the other side to a kinesin, and then we start pulling them apart. Okay, so we're physically pulling them towards the pole, but then at the same time, they're being sort of pushed away too. So there are two things that are happening. So you're having a push from this side against that other chromatid, or kind of from the center line here, they're getting pushed this way, and then from here, they're pulling them towards it. In the end, it doesn't really matter. What's happening is your sister chromatids are being separated and then pulled to each side. But they're getting pushed from one side and pulled from the other side so that they end up at the poles. Okay, and so if you want to dig super deep, I mean, you can see it's really complicated. There's all kinds of things going on because it's not just one big old honking thing attached to a sister chromatid at the kinetochore and pulled. It's a massive complex that has dinins and kinesins and chromokinesins and a bunch of things that are just entirely devoted to this process that occurs during mitosis. That's their whole job. And so something else to think about is that these things are all proteins that have to be made by transcription and then translation. So that sounds like a lot of extra work. It is, and you got to plan to do it before you can actually do any of this stuff. You can't do any of this if any one of these things is broken. Something's going to go wrong. So that should be absolutely terrifying to you. Now, as those motor proteins occur. So we need to kind of dig a little deeper into some of the signaling that's actually going to go on. Underneath is the wrong word, but that's happening simultaneously to this process. I'm so sorry. Let me put this down here. You can see what I do to actually do my references. There we go. Stick that down there. Now, the whole point of this should actually look kind of familiar. So having CDKs and the amount of CDKs. So we saw this with the cell cycle, that the different amounts of CDKs and cyclins, it depends which CDKs are there and which cyclin-dependent kinases are there, and that controls the cell cycle. Well, we see that, well, here's G1 going to S to G2, and we see our cyclin-dependent kinases. It's referring to some other things here. But you can see we have, after our checkpoints, here's our prophase, metaphase, we've got our anaphase initiation, anaphase A, and then anaphase B, and then we're going to head to telophase, right? If you want to read this paper, it's really interesting. It talks about PP1 and PP2 here, and how they actually function to stimulate this actual separation and the movement from anaphase into telophase. Really, really interesting. We're not going to go into too much depth here. But separase, there's that separase, right? So if I back up just a little bit, there's separase right there. So the presence of an active separase goes up when we have the initiation of anaphase right here. Right? So there's our metaphase to anaphase transition, and that's what's happening right here. This is your metaphase to anaphase transition, and look at that. That blue line matches. Whew, good thing it matches up. Oh, that would be really embarrassing if it did. Oh my gosh, there's a lot going on here. Now, I'm only showing this to you because I wanted to show you something, the kind of thing that I can put on an exam, where I would give you a picture like this. So this paper that this actually came from, which it's from this same paper. The picture came from that paper, and it's talking about the complexities of anaphase initiation, that movement from metaphase to anaphase. I'm not super interested in you memorizing all this stuff. What I am interested in is if I showed this to you and asked you questions, you could answer it. So if I said, what happens if I break PLK1? Well, looks like it's a kinase that's phosphorylating, and thus it would prevent this furrowing signal. You don't have to know what that means. It basically means that the microfibers or the microtubules they cross over a little bit, and you initiate the furrowing or the separation of anaphase with that. But that's unimportant. If I said, if you break this, what happens? Well, this is broken. Right. That's actually pretty straightforward, isn't it? Anyway, so this is also to just kind of either terrify you or awe you with the complexity of life. There's histones there, so it's involved with histones. Oh man, I fell down very deep the rabbit hole. Now, let's keep going. So we've gone through prophase, prometaphase, metaphase, anaphase. We're on to telophase. We're going to start fixing all this stuff. So we're going to assume that we've separated our sister chromatids. They are now in new cells and our new daughter cells. And now we need to just start fixing the problem. So we're going to reform our nuclear membrane. You'll see the subnuclear compartments, so the nucleolus being one of those will start to reform. This is just like if you're looking at our microscope, that's what you physically see. That's why we list that. And so there's the nucleolus there. The spindle fibers start to kind of dissolve. So I said dissolution of spindle fibers. Really, it's not just that. Spindle fibers are incredibly complex, as I mentioned. And they're just going to kind of become part of the ether of the cytoskeleton. And then your chromosomes are going to, they mention here decondense, which isn't actually a word, but I'm going to say decondensation. So they start to get loose again so that we can start doing transcription. Or maybe I need to double again, so I need to have it nice and loose so I can actually go through the S phase and then, keep on doing my thing, okay? So cytokinesis is actually, so sometimes you'll see cytokinesis listed after telophase. It's really the process, so we've been dealing with chromosomes, right? That's what mitosis is really talking about. Prophase, metaphase, anaphase, telophase. What's going on with the chromosomes and what do I have to do to make the chromosomes? Cytokinesis is more of a zoomed out picture, what's actually happening to the cells and especially the cytoplasm. That's a good way to look at it. And so cytokinesis is looking at, okay, well, I had one cell. What am I going to do to get two cells? Well, I have to double everything and then split down the middle. That's what we see with cytokinesis. And that actually starts happening earlier than after telophase. We actually terminate it. You have two final daughter cells after telophase, sure. But you have that process kind of happening simultaneously to what's going on with the chromosomes. And so what we actually see here In animal cells and plant cells, I mean, they always separate these out, but it's similar. It's just the cells are different. So if you have an animal cell, you have actin, which is part of your cytoskeleton. It's obviously more complex than this. I already went into so much detail with all this crazy junk that I decided not to go into too much detail with the furrow, though I do have one diagram. And so when you have that cleavage furrow occurring, notice that the end products of telophase can be occurring, or even anaphase can be occurring while this occurs. Because once I've got my chromosomes separated out, go for it. We want this cell to be done as soon as we can, right? In plant cells, you start forming that cell plate, so the cell wall starts to reform, but it's the same idea, right? You're going to separate those two cells, okay? Now, I said, ooh, I want to know if there's a pathway. Of course there is, associated with cytokinesis, and you have to polymerize your actin to actually form that furrow, right? So we're here forming this furrow. And that occurs by, oh, surprise, surprise, a bunch of kinases. Look at all these kinases. There's a kinase, these are all kinases, and the kinases are phosphorylating each other. It's just this crazy thing, and then lo and behold, look up here, P27 and P21, those are tumor suppressor genes, or tumor suppressor proteins made by tumor suppressor genes. And those can block this entire process, which is very, very interesting. Wow. So you're going to start seeing some repeats in pathways of things like AKT has shown up before. There's a heat shock protein here. Yeah. So and there's P38 alpha, which is just a subcomponent. But anyway, it gets complex. If I would give you a picture like this on an exam and then ask you questions like, oh, if I disabled cofilin, what does that do? Oh, well, if cofilin doesn't get phosphorylated, then I can't actually polymerize my actin. What would that do to the cell? Well, you would never be able to form a cleavage furrow, and you would end up with these weird bizarro cells that are like two separate cells, but they're still attached, and it just gets confusing. So, anywho, move on. So that is the end of mitosis. We've gone from prophase, metaphase, anaphase, telophase, cytokinesis. We've done it all. It's great. Let's talk about meiosis. Okay, so in meiosis, that's where we need to make gametes, okay? Basically, every eukaryotic organism undergoes some form of meiosis. They don't necessarily have to, and so you can have, I mentioned it down later on, but you can have some creatures undergo parthenogenesis. So that's asexual reproduction in eukaryotic organisms. And so what does that mean? Well, some fish can do it. There are a couple animals that can do it. It's relatively rare. Some of the lower order organisms, like rotifers, can do it. Anyway, the lower order stuff like that. They can basically just do, they can create new living offspring through essentially mitosis. So if one of these cells went off and then formed an entirely new creature, that's parthenogenesis. But usually we're not doing that because we don't want our offspring to, I'm going to use a double negative. We don't want our offspring to not have sexual reproduction. We want them to be a mix of a mother and a father because that's going to increase the amount of genetic diversity in our offspring, which is a good thing as a species because the more diversity we have, the more we can resist changes or selective pressures that are applied to us. And so we need to talk about meiosis because it's really, really important. So in mitosis, which I apologize for the quality, but this is honestly the best version I could find. In mitosis, we go from having, so N is our number of chromosomes. So we go from 2N to 2N to 2N to 2N. So we're always maintaining one from mom and one from dad. Okay. During meiosis, we're going to make a bunch of gametes that basically what we're going to do is if we had, let's say we went through S phase, right? So let's back up a little bit. So we go through S phase. Let's look right here. Okay. So this is where we were talking about it. So we've gone through S phase. So we have a copy from dad, a copy from mom, copy from dad, copy from mom, right? If I took this 1, 2, 3, 4, because I have 4 sister chromatids here. If I took each of those, I'm sorry, let me look at it here. So let's just look at this one. Sorry, I was misspeaking. So if I have 1 here, 2, 3, 4, right? So I have a homologous chromosome, right? These are, let's say, chromosome number 1 in a human. Okay, so I have 4 sister chromatids that all code for chromosome 1, right? Normally it would just be a single one like this, but I doubled them during S phase, and I have two of these, one from mom and one from dad. And if I separate each of those out, so this one, two, three, four, if all of those went into their own individual cell, that's meiosis, okay, where I separate out my sister chromatids. Now we're going to do some stuff to them first before we get there. We need to go through basically 2 rounds of mitosis, which we call meiosis, and we need to increase our genetic diversity. Okay, so we're going to do 2 big things. We're going to have crossing over occur, and we're going to do independent assortment. Talk about that a little bit. This is from Wikipedia. It's honestly the best one I could find for what we need, because a lot of this is going to seem really, really similar. So our steps, our phases are going to match up with what we just talked about. Hence why I'm not going to go into extreme detail with prophase 1, metaphase 1, anaphase 1, telophase 1, and 2. We don't need to do that. Like, we're just going to basically see similarities happen, and then I'm just going to address some of the different things that happen, okay? So prophase 1, same stuff's happening. The nuclear envelope's going to break down, but we're going to get crossing over happening, okay? So let's talk about crossing over. So crossing over is when I have my homologous chromosomes. So in this case, this would be like chromosome 1, chromosome 12, chromosome 21, who cares, if we're in like a human. And remember that we have 1 copy from mom and 1 copy from dad. Okay, so let's zoom in on one of these. So let's grab this guy and we're going to zoom in on him. So here's a copy from mom and a copy from dad. Okay, and then these are representing the alleles. Okay, but notice they're the same, right? Because we started out, we had one of these sister chromatids from mom and one of these from dad, and then we doubled them during S phase, right? So we have 1, 2 from mom and 1, 2 from dad. And what can happen is, remember, the locations of the genes are the same. The actual genetic structure, or the orders of the A's and the T's and the C's and the G's, can be different, but they still code for the same thing. So eye color would be A, let's say, and hair color could be B. So I could have brown eyes and blue eyes, but they're still eyes. They're still in the same location for eye color, I guess. And so what happens during crossing over is when they just swap. They kind of put their little arms over. Oh, it's so cute. They're holding hands, and then they swap. Mistakes can happen here. People ask that a lot. Of course, we're going to talk more about that when we have chromosomal abnormalities. This is where you can get a lot of weird things happening. But usually problems don't happen, and you can get a nice crossing over event. And that's super cool. So that's going to increase our genetic diversity, because you can see here, we now have a sister chromatid from mom that looks different from this sister chromatid from mom. Okay, so now those are 2 different things. Yeah, they share some genetic similarities, but they're now separate. So if I took this chromatid and mixed it with a new sex cell from another person, okay, so let's say this is a female, so this is going to go into an egg, and then a sperm from a male is going to come along and match up with this one, that would create a completely different human than if I took this one and put it into an egg and matched it with a sperm from somebody else. Okay, so that's where we're getting some more diversity, which is awesome. Okay, that's what we want. So that's crossing over. Now let's back up. So that occurs really, really early on during prophase 1. That's when crossing over occurs. The second way that we get some diversity in, which is called independent assortment, happens shortly thereafter. That's during metaphase 1. It can feel tricky, but it's not. So instead of lining up along this axis, okay, so we've got our metaphase plate, it's happening here. What we used to do is, let me pull up metaphase here. So what we used to do is line up our homologous chromosomes, they're kind of end to end, and we're going to pull apart the sister chromatids. That's not what we're going to do. What we're going to do is going to take, like, these 2 match up, and I'm going to put them next to each other instead of top to bottom, okay? So there we see this red one and this blue one. Those are homologous chromosomes, not sister chromatids, right? The sister chromatids, we had crossing over occur, and they're still attached at their centrosome, at the kinetochore. Okay, so we're going to pull apart our homologous chromosomes, and then we're going to pull apart our sister chromatids, and that's the second half of meiosis. This is called independent assortment because I could just imagine with me, see how we've got a red one here, let's say that's from mom, red one here from mom, blue one from dad. Okay, so I don't put all of the mom chromosomes on one side and all of the dad homologous chromosomes on this side. Okay, it's kind of random. They just show up on either side. Okay, so that's independent assortment. I don't have to have all of mom go to one set of cells, and then all of dad's stuff go to another one. They're sort of mixed. And so that's where I'm going to get, I could have, well, it's dealing with separate with the homologs. But does that make sense? I hope it does. If you have questions, please ask. Come to one of the video sessions that I have to ask questions about this. Honestly, this is something that I struggled with a lot. And so just ensuring that I understand what's physically happening. I'm not pulling apart my sister chromatids. I am pulling apart the homologous chromosomes, and it doesn't matter which parent goes on which side for which chromosome. It doesn't matter. The cell doesn't care. Okay? Anaphase is not that different, except instead of pulling apart the sister chromatids, they're still stuck together, so we're just going to kind of pull them. I always like that. They're like, ah, and they're like getting pulled apart. And then we are going to reform a new cell. So you get these weird like cells that have, they just look weird. But you're going to form two new ones because it's easier to then split this cell and split this cell than trying to sort of form two or four cells out of like one. I don't know. It just gets complicated. I do have a picture of independent assortment. I forgot. I have a picture here. So if you need to look at this and just kind of look at it more, but basically, so look here. Here, I've got my Metaphase plate going down the middle here. If I put so dad's like the blue one and mom's the pink one, okay, just to be, gender stereotypical. And we've got all of dad's chromosomes on one side and then the homologous chromosomes from mom on the other. And then I'm going to split them. So these are all for dad, and then I'm going to pull apart the sister chromatids here. Okay, so that's meiosis II is basically what's happening here. Okay, so and then I ended up with all of dad, all of dad, all of mom, all of mom. Okay, awesome. I don't have to do it that way. I could have split it up. So let's put one of dad's chromosomes on one side and another one of dad's chromosome, or one of mom's chromosomes on this side. And so my new daughter cells can be a mix of dad and mom. Okay, so that is independent assortment. And you'll hear that when we talk about Mendelian genetics and non-Mendelian genetics, the law of independent assortment is going to come up again. But you can see here, this is really nice because it's talking about, we have Metaphase I, where this happens, and then during Metaphase II, we have that second lineup. Metaphase II, okay, which occurs during Meiosis II. Okay, so here's Meiosis I, okay, little ones. Meiosis II is essentially like mitosis. Okay, so we're going to have our chromosomes condensed down. They line up along the metaphase plate, and we pull our sister chromatids apart. Okay, so it's almost identical to mitosis. So if I asked a question like a common question I've seen before, which of the meioses, meiosis I or meiosis II, is more similar to mitosis, and you would say meiosis II. Right, because you're pulling apart the sister chromatids. Okay? So that's the trick there. So we end up with now each of these cells. So we're left with 4 daughter cells, and they only have one sister chromatid from basically grandma, sort of. And so this is going to then become like one egg. If you're forming ova, then you tend to disable the other 3. Okay, you don't always do that, but sometimes you do. So you're left with one egg, basically. That gets complicated. Or if you're male and you form male gametes, then these would each become a sperm. So they become flagellated, and yeah, awesome. So that's meiosis, and you're left with a single chromosome, as opposed to, instead of being diploid, you are now haploid. Okay? You are now N instead of 2N. A couple links that I put on here. This was to remind me to talk about parthenogenesis, which we did. So there are a couple things. So I want to show you this cells alive thing. So hey, look, he's still walking. So cells alive, yes, they use Comic Sans. Don't hold it against them. I apologize for that. It's my least favorite font on the planet. It's a nice little click-through-y thing. So you're going to hear my mouse click a few times because I can't do it with the keyboard. But what it does is it's going to take you through each step of meiosis. So if you really need some help, which I always do, I always need extra help. Meiosis here, you can click through. So we're entering prophase 1. So we have condensation. And we know during prophase 1 is when crossing over occurs. So let's see. Oh, there it's happening. Okay. So in this case, they're going to have, let's say, chromosome 1 and chromosome 2. In humans, there would be 23 of these. And so chromosome 1 lines up. This one's from dad. This one's from mom, and we're going to see crossing over occur. And there it is. So now we're left with these new chromosomes that had crossing over, and they had it happen on this chromosome as well, right? So we saw that little swapping there. That's also something in the lab for this class that you're going to encounter. We can do map distance with the fruit fly lab. And so you can actually calculate based on how much homologous recombination occurs in the offspring. You can use that to figure out how far apart genes actually are on the chromosome, which doesn't really matter anymore because you can just sequence it, but hey, it's kind of fun. And so that's where, that was during prophase 1, so we're kind of moving on. Now we need to get our metaphase happening, and remember, in metaphase 1, this is where we have independent assortment occurring. So this could basically independent assortment would say, see how that's kind of, they're falling this direction, right? So the pink one's going to be on the bottom and the yellow one's on the top, red's on the top, blue's on the bottom. It doesn't have to be that way. Blue could have been over here. Pink could have been over here. It doesn't really matter, or whatever combination there is. And so you can imagine in like a really simple organism like that only has 2 chromosomes, I mean, yeah, like there's not a ton of diversity being incurred. But if you have 23 pairs, that's a ton of diversity. And you could just send one from mom and one from dad into either one. And so you were just going to see that happen. So there's independent assortment just happened, and you keep clicking through. They separate, we go through telophase, reforms up, and now we're going through basically meiosis II is just mitosis again, right? So we condense down, we're going to pull our sister chromatids apart, and then we form all of our daughters or gametes, okay? We're left with that. So I have a link there for that, so if you want to click through it. I sat there and played with it for a while because, let's be honest, I need to review some of this material sometimes. So also, here is a link to a awesome meiosis video. It goes into great depth, especially for like a YouTube-y kind of video. It's really, really good. It's from McGraw-Hill, which is a textbook company. I'm sure you have textbooks from McGraw-Hill. It's from 2010. The only videos that I could find, it's one of those, it's like an interactive video that they have. And so the ones that are out there aren't from McGraw-Hill. They're like stolen, usually by like Indian dudes will steal it and put it on there. It doesn't have anything, it's not a judgment on the Indians. It's just, thank you for doing that, because now my students can watch it. And it's, otherwise, this isn't even behind a paywall anymore. It just like doesn't exist online. And so thankfully somebody saved it. And there's a bunch of Indian dudes that and ladies that will put awesome videos up. And they're really helpful. This is the higher quality one. But that I wanted to give a shout out to where it actually came from. But it's 10 years old now, so I hope they're not too upset about it. Anyway, but that's a really, really good video. I would suggest watching that. And if you're interested in understanding more about evolution, how different organisms have evolved, because meiosis isn't always identical in every creature, or why in the world do we even engage with this process of meiosis? It's super complicated and lots of mistakes can be made. Go ahead and read this article. It's free and available. It's huge and awesome and has really good diagrams and goes into amazing depth. So go ahead and check that out.